Okay. Thank you for coming on our early morning session. It is midnight in Japan, anyway. But I'm Masayuki Nomura from Asahi Kasei Corporation, a Japanese chemical and material company. And I will be chairing this session as a member of AIRM. Aim of this session is to share the Japan's latest situation, the industrialization platform. Okay, the session is a very tight schedule, so let me start this session. First speaker is, next slide, please. Okay, first speaker is Mr. Tetsuya Tanaka from METI, Government of Japan, for opening remarks. Mr. Tanaka, please.
Good morning, everyone. I'm Tetsuya Tanaka from METI, Ministry of Economy, Trade and Industry. It's my great pleasure to welcome you here today. Thank you very much for participating in the Japan session today. METI has been committed to actively commercializing regenerative medicine. Japan is a promising market. As you probably know, Japan has already become an ultra-aging society ahead of the rest of the world. Changes in the demographic structure are already affecting Japan's society and economy in various ways, even greater impact anticipated in a broader range of fields in the future. In light of this social background, Japanese government is now revising healthcare strategy, which is a 5-year plan. For the next healthcare strategy, which is effective from 2020 to 2025, we will put more focus on preventive medicine, regenerative medicine, and cell and gene therapy. In addition, this year, the Japanese government formulated the bioeconomy strategy aiming at realizing advanced bioeconomy society by 2030. It's been 10 years since the Japanese government made the last strategy. Secondly, let me briefly mention R&D activities for regenerative medicine in the public sector. The Japanese government has been promoting integrated medical R&D activity from basic R&D to R&D focused on practical application. The Office of Healthcare Policy in the Cabinet is a control tower in the medical R&D, coordinating budget funds and projects of related ministry. MHLW, Ministry of Health, Labor and Welfare, MEST, Ministry of Education, Science and Technology, and METI are 3 major ministries, and AMED, Japan Agency for Medical Research and Development, is a governmental finance agency for medical R&D. 3 ministries and AMED work together, sharing the same R&D goals and plans for regenerative medicine and gene therapy. The total number of government R&D budget for regenerative medicine and gene therapy is approximately $140 million this year. It will be $200 million next year if the Diet approves it. Thirdly, it's about rules and regulations for regenerative medicine. We have 2 major acts for regenerative medicine. One is Regenerative Medicine Safety Assurance Act. The other is Pharmaceuticals and Medical Devices Act. The Safety Act regulates medical institutions that provide regenerative medicine. Any medical institution has to submit the regenerative medicine provision plan to the government before they do clinical research or medical treatment at patients' own expense. As of the end of April, about 4,000 plans have been submitted to the government. The Safety Act also regulates CPFs, cell processing facilities. The Act enables medical institutions to outsource cell culturing and processing to the facility run by private companies. The private companies need to get approval from the government when they run CPFs. Although medical institutions have their own CPFs, the number of CPFs owned and operated by private companies is now about 60 in Japan. 7 CPFs located overseas are also approved. Regarding Pharmaceuticals and Medical Devices Act, we have a specifically ARI approval system for regenerative medical products. The Pharmaceutical Act allows us to give conditional and term-limited marketing approval to regenerative medical products as long as the efficacy can be assumed and the safety is confirmed. So the pharmaceutical company can provide products as early as possible in the domestic market. I think Dr. Yamada from Anjus will tell you more explicit story later because his product got this special approval last month. In addition, the government implements Sakigake designation system. The Sakigake means first track in Japanese. The Sakigake first track system aims at shortening pre-market review period for innovative medical products, including regenerative medical products. Under the Sakigake system, the designation process will be prioritized, and the pre-market review period will be cut in half, about 6 months relative to general new pharmaceutical products. Although some issues are still being discussed, including drug prices for regenerative medical products, Japan's regulatory framework for regenerative medicine and its product is well prepared and business-friendly. Dr. Maruyama from PMDA will give you more information about rules and regulations in Japan later. Before I hand over to Dr. Shikata from Pharm, I'd like to briefly mention METI's perspective on doing business for regenerative medicine in Japan. Thanks to Dr. Yamanaka receiving the Nobel Prize in 2012, iPSCs, induced pluripotent stem cells, are well recognized among Japanese people. Our study showed that more than 90% of the Japanese people have heard of iPSCs, which is the highest rate of public awareness in any other major countries. In the United States, it's only about 30%. More than 80% of the Japanese people are in favor of regenerative medicine and gene therapy. As I mentioned earlier, METI has been committed to commercializing regenerative medicine and gene therapy. Specifically, METI has invested more funds on commercialization of manufacturing technology for regenerative medicine and gene therapy. Also, METI often set up informal meetings with leaders from industry and academia for new rules and regulation in order to disseminate new medical technology and create new healthcare businesses. Currently, we have intensive discussions about how we steadily provide cell sources for regenerative medicine in Japan. Next week, we have BioJapan in Tokyo, which is the biggest biotech event in Japan. METI initiated this event to promote international collaboration. Come and visit to Japan, a promising market for regenerative medicine and gene therapy. I think Ms. Tsurumaki from Novartis will tell us an inspiring story about CAR T therapy in the Japanese market later. So once again, I'd like to thank you for coming today to this Japan session. I hope that this event will be meaningful and productive for everyone here. Thank you very much.
Thank you for Tanaka-san. Next speaker is Dr. Yoshitsugu Shitaka from Astellas Institute for Regenerative Medicine. Shitaka-san will talking about the evolving market and industrialization platform. Thank you.
So thank you for the introduction. I'm Yoshitsugu Shitaka, Vice Chairperson of Pharm and also the President of Astellas Institute for Regenerative Medicine. Next, please. Today, I'm going to talk about Pharm first, and then which is followed by the Pharm's effort for international standardization. And lastly, I'm going to touch upon the industrial platform for regenerative medicine in Japan. Next, please. So Pharm was established in 2011. It is a Regenerative Medicine Industrial Association in Japan. And Pharm consists of more than 250 members coming from various industry sectors: biotech, pharma, machinery device, and chemical materials. And okay, thank you. And logistics, insurance consulting. And our mission is to accelerate industrialization of regenerative medicine in Japan. Sorry. Okay. This shows the org chart of Pharm. We have 14 committees. Committees in red. They're working on the common industrial challenges, such as standardization, regulation, and pricing. And committees in green, they are related to our organizational operations. And bottom three, they are involved in the national projects. For instance, we are working with AMED to standardize the manufacturing criteria or safety criteria. And you know that ISO is an international standard setting committee, and the Pharm organized the Japan Miller Committee and have made a significant contribution to the operation of ISO TC 276. Board of directors consists of 20 members. Representative director chairman is Kenichiro Hata, Japan Tissue Engineering. He has been assigned as chairman since June this year, 2019. And we have 3 vice chairpersons, Dr. Osakabe, Dr. Suzuki, and myself. And I also serve as a representative director. Okay, and next is our efforts for international standardization. Before that, we developed the Pharma Vision 2025 this year. Toward 2025, Pharm leads the global regenerative medicine market in collaboration with other industrial groups. And also, Pharm commits to providing reliable information to stakeholders and also delivering proposals that create change. And also, Pharm continuously strives to create innovation. This innovation includes not only technological innovation, but also the non-technological, so-called marketing innovation, including supply chain, pricing, sales channels, etc. We have concluded more than 10 MOUs with overseas industrial groups, such as AIRM, CCRM, UK Catapult, and also the scientific societies. Today, I'm going to share with you our initiative with Asian countries for international standardization. APAC-AIRM, which stands for the Asia Partnership Conference of Regenerative Medicine Associations. We kicked off this initiative in 2018 with China, Korea, Taiwan, India, and Singapore. Mission and goal of these activities, APAC-AIRM, is to expedite the development of regenerative medicine products for Asian people by promoting the harmonization and optimization of the regulations on AIRM products among Asian countries. And also the important other goal is to maximize the opportunity of Asian market and create a consolidated, sizable regenerative medicine market, which is comparable to those in North America and Europe. Again, one of the major goals of this activity is to promote regulatory harmonization of RM product and reducing the time and cost required for clinical development. So we agreed in the second APAC RM, which was held in April this year. We agreed to develop the working groups to discuss, to harmonize, for instance, the test items for non-clinical studies, quality of data, the way of data sharing, and also the other working groups. They will work on the to discuss what comparability studies should be, and also the align the biological materials requirements from each government. So we'll have a in-person working group meeting this month in Yokohama, and the third APAC RM will be held in next April. And lastly, I'd like to share the current status of platform in Japan. But before that, I'm going to briefly touch upon the market status in Japan of regenerative medicine. By 2018, 5 RM products were approved. JACE and JAC were developed by the JTEC, and also the heart seat from Thermo. This is an autologous myoblast seat. This program is currently under conditional and time-limited approval status. And TEMCELL by JRC, this is an allogeneic MSC product, was approved in 2015. And Stemilac being developed by Nipro, this is an autologous MSC product. This is also under the conditional and time-limited approval status. And as Tanaka-san mentioned, this year, 2 new RM products were approved in Japan, Collagen by Anges, and Kimuria developed by the Novartis. So Yamada-san and Tsurumaki-san will give you talks on these products later on in this session. And this is a snapshot of a Japanese clinical trial of regenerative medicine. As you can see, 2018 was very productive, fruitful year for us. About 20 regenerative medicine products initiated their clinical trials in Japan. Oncology accounts for about 50% of trials in Japan in RM space. And half of the programs are strongly driven by biotech companies. And this slide summarizes the manufacturing and the clinical trial operational platform in Japan. For example, the Kyoto University and the National Center for Child Health and Development, they provide us the clinical grade iPS cell line and also the ES cell line. And a Foundation for Biomedical Research and Innovation, or Kanagawa Life Innovation Center, they are playing very important key roles in biomanufacturing space. And also some of academic hospitals, they successfully developed the clinical trial operational platform knowledge experience in Japan. This is the example of biomedical research and innovation at Kobe. They provide us various services. For example, the safety test service and research for cell specification and cell manufacturing, process development. And also they give us a consultation service for cell manufacturing, and also the development of actual process. And also they provide supply CTM and commercial product to us. So, and this slide table summarized the comparison of regenerative medicine industrialization platform. Stem cell bank, cell therapy manufacturing, cell therapy translation in Japan, US, and EU. So in Japan, again, we successfully developed a basic industrialization platform in these categories. And as I mentioned, the number of clinical trials are getting increased. And those are strongly supported by these platforms in Japan. So in conclusion, as Tanaka-san mentioned, innovative Japanese regulations have been enacted and product creation is being promoted. And in Japan, our robust platform accelerate the industrialization of regenerative medicine at this moment. And Pharm, we will promote regulatory harmonization and international standardization in the field of regenerative medicine. Thank you very much.
Thank you for Shitaka-san. Today, so no Q&A session. Sorry. Next speaker is Dr. Yoshiaki Maruyama from PMDA. He will talking about the update of regulatory landscape of regenerative medicine.
Good morning, ladies and gentlemen. And thank you for the very kind introduction for me. And then this is the first time participate in this MESA cell and gene meeting. So, and then this year we moved to the from the La Jolla to here. So I'm very exciting to be here. And then last night we had a good dinner in here, that over the restaurant. And then I think this meeting is, that conversation with everybody. We also very exciting to communicate with sponsor or developer to this meeting. So as I mentioned before, this session, no Q&A, but I think my presentation is very short. So if have a time, I would like to take some question from floor. Okay. So my presentation is update for the regulatory landscape of the regenerative medicine. So. Okay. I prepared the 2 points. One is the current situation for as I mentioned before, we have 2 acts, the Safety Act and then the PMD Act. So I will be introduce actual product for these topics. And the second point, I think in the USA, now the gene therapy development is very increasing now. So actually in Japan also, same situation. So that's why I briefly introduced our gene therapy regulation in the later. So this is, I use many times to this cartoon. So we now in Japan, 2 acts regulate the regenerative medicine. So one is right side is act on the safety of the regenerative medicine. So this is regulate for the medical care or medical practice, or clinical research. And then the left side, the PMD Act. The PMD Act is regulates the product for the regenerative medicine. So this is originally already the 5 years ago that act is enacted. That time already medical practice using the cell therapy is existing. So, but we don't know that who and then what kind of cell therapy is doing that time. So that's why this act required to the registration to the ministry. So that's why we now recognize to the what kind of cell therapy is who's doing in Japan. Okay. So that registration, I mean that the hospital clinic must be submitted the plan to the ministry, but this plan is reviewed by a committee. So, but we in the government is classified this 3 category: high risk, middle risk, and then low risk. Then the discussion for the committee is different, depend on the class, these class. So class 1, high risk, is very manipulated cell therapy things. Like example is iPSC derived cell therapy or allogenic cell therapy product. And then class 2, class 3 is autologous cell, but homologous use and then non-homologous use is different for the class 2 or 3. Okay. So high risk submitted to the plan to the special committee or MHW, and then reviewed by this committee, and then the ministry is asking to the opinion to this special committee, and then if the plan is okay, then they can try to the like clinical research or medical practice or this cell therapy. So the special committees number of the special committees is 52, and then the regular committee is 97. So this is a number of the approved plan. So I think the class 1 is here. So already 20 clinical research plan is approved. And of course, I think that the medical care or medical practice is there. That means good for, I mean that this very manipulated high-risk product is not only so this plan is mainly reviewed for the safety, not for the efficacy. So that's why if the safety is okay, then the plan is approved. So, but for the right now, so everybody think about how about the efficacy for this plan. So right now, actually the government or society of Japanese Society of the Regenerative Medicine is discussing about how to collect the efficacy data for these cell therapy for the medical care or medical practice in the actual real world. So this is a number of the plans. So the beginning of the after the enacted in the this act, the class 3 is immediately we can recognize the number of the plant, and then 5 years the trend is like this. So next slide, I show you that actual approved plant for the class 1 for the clinical research case. So one is iPSL for the spinal cord injury is Keio University. So this is approved in the February this year. And then second product is again this is a iPSL derived cornea transplant from the Osaka University. So this plan is approved in the March of this year, and then already conducted in the transplant in the first patient. And then Dr. Nishida already comment on that the first transplant cornea disease for the data for this transplant. So, but every time this report in the approved in this product, some people asking the actual real product is approved. So I mean the PMDA reviewed to this product and then approved in the commercialized to the post-marketing phase. But this approach means that just plan for the clinical research is approved. So please do not confuse into the approved mean. In the two acts, Safety Act and then PMD Act is different. Okay. So next, I will introduce the approved product under the PMD Act. But before, this slide is actually 2 years ago. We have a panel discussion for the regulator session in this MESA meeting. I just slightly modified to this slide from that there. And then, so all 3 regions has a early access scheme, like PRIME, the EU, RMAT, or the US FDA, and then the PMDA has a PMDA, MHW has a Sakigake designation system, and then conditional and time-limited approval system. So this is for the strategy of Sakigake. 2 years ago, I presented the Sakigake system in this session, so I don't mention anymore. But and also I wrote the article for the Sakigake or conditional time-limited approval in the Bio Insight. So if you interested the detail for this system, please access to these articles. So now 14 product is designated in the Sakigake. Then I showed the two slides. This is fourth round 2019 this year. Is designated two products. One is OBP 301 from the oncologic biopharma. This is for the oncologic virus for the esophageal cancer. And the other is Sambio, the SB623, is a bone marrow derived mesenchymal stem cell for the traumatic brain injury. So last month, end of August, I think. So fifth round for the Sakigake designation is announced by MHLW. So if you interest, if your product is want to granted this Sakigake designation system, please contact to the MHW or PMDA. So I think the end of the November is the deadline for the applied to this fifth Sakigake designation round. So next, this is again I use many times this slide. So this is a scheme for the conditional and time limited approval of a system. So now in Japan, seventh product is approved, but and then 3 of them is granted for the conditional and time limited approval. And then one is the Stemirac, the autologous mesenchymal bone marrow derived mesenchymal stem cell for spinal cord injury is approved in the last year. This is actually a Sakigake designated product in the first round, and then of course, again the conditional and time limited approval product. So this product is only a single arm clinical trial data, but we think we reviewed that, and then we think that efficacy is expected in this product. That's why we can approved in the conditional and time limited approval system. So, but in the Nature editorial is there is some issue for the very early clinical trial data to approve for the post-marketing phase is sometimes very challenging. And then the Nature also pointed out it should be conducted randomized clinical trial for to the approval in the product. But again, we think the evidence level is randomized clinical trial data is very good. But sometimes, like especially these autologous cell therapy product is collecting the clinical data is takes long time to confirm the data. So that's why this system is enacted 5 years ago. So it's for the whole Japan side, it's okay for this still exploratory data, but after the post-marketing phase, they try to confirm the efficacy to setting external control to compare to the efficacy. So we are waiting to the after the collecting data in the post-marketing phase. And then this is again ANGES for the HDF plasmid for the ultra healing for the clinical limb ischemia patient. So Dr. Yamada will be presented it later. This is also the condition and time limited approval product. Oh, sorry. And then this year, the Kimraya for the AIRM, CAR-T for the AIRM, and then diffuse large B-cell lymphoma. So Dr. Tsurumaki will be present later for this product. Okay, so this is an update for the our two regulations, two act, and then let's move on to the update for the gene therapy regulation in Japan. So as I mentioned before, this is Japan's case for the trend of the gene therapy development. So left side is a consultation number, and the right side is a IND number. So in the act for the PMD, the PMD act, the gene therapy is actually different category compared to the United States. Gene modified cell is a cell therapy product. So, but this slide shows gene cell therapy is including gene modified cell like CAR-T cell. I adjusted the United States version for this number. So I think the, every year, gene cell therapy, the consultation number and the IND is slightly increasing, like as United States. Then we recently revised two guidelines. One is a guideline for the gene therapy clinical research in the February this year, and then second is a guideline for the assuring the quality and safety of the gene therapy product. This is very recently revised in the guideline. So if your product is a gene therapy product, like in vivo gene therapy product, or ex vivo gene therapy product, please refer to these two major guidelines to your research and development phase. And then let's move on to the to introduce the new consultation menu. This is the Cartagena issue. Why I say issue? Because time this Cartagena domestic law is very specialized in Japan compared to the United States. We need to do the protocol to the type 1, type 2 Cartagena law is mandatory before starting the clinical trial or clinical research. So, but sometimes it takes a long time because most of you don't know how to prepare to this document. So that's why we, in the PMDA, announced the new consultation menu for this Cartagena domestic law. So this is the purpose of the law. I don't mention anymore. So just smoothly implemented the protocol by taking measure to regulate the use of the living modified organism. And then this is the scope of the law. I think the in vivo gene therapy, the genetically modified BIOS is, of course, strictly need to the scope of this law. But genetically modified cell therapy product is out of scope. But if remain the BIOS to modify the cell like CAR-T or iPS cell using the BIOS, but that BIOS is remained in the cell, still need to this is the scope of the law, because remain the BIOS in the cell needs to be scope of this law. So if your product is a ex vivo gene therapy product, we ask the BIOS is still there or not. If the BIOS is not detection under the detection limit, then you don't need to prepare this document, the Cartagena law. But if the still BIOS is there, you have to prepare the protocol for the Cartagena domestic law. So what Cartagena law need is just you should prepare the procedure of the type 1 use. I mean that the use of the living modified organism without preventive measures against their disposal into the environment. So that means please file the document or the to the environmental risk assessment, and how about the risk assessment for the third party, like not only the patient, but how about medical practitioners or that family. So this need to the assessment, and then prepare to the how to use this product to in the actual phase. So the procedure need to the how to store this product, or how to transport this product, how to dispose to the contaminants or the BIOS, and then shedding, how to collecting the shedding data in your clinical trial phase, or like that. So not so difficult, I think. And then we don't ask to the shedding data before starting clinical trial. We know there is no clinical shedding data before clinical trial. So, and then the type 2 is this is for the manufacturing site. If you use BIOS in your process, then need to the type 2 use procedure of the type 2 use. And then, so the type 1 use is needed to approve by Ministry of Health, Labor and Welfare. And then this is just needed to confirm for the, from the minister. So again, new consultation menu is started on this April. So if you struggle to prepare this documentation, please contact the PMDA. And then last, the PMDA has a science board. So the science board is discussing about when our reviewer struggle to how to review this issue, specific issue. Then we previously discuss in the science board and then making the concept paper of these two. One is homogeneity, and then other is basic principle for the quality assurance of the cell therapy product. And then last year, we started to the gene editing issued. Already conducted many time to meetings, and then already drafting paper is the concept paper is already drafted. I think hopefully this year or next next year we showed you the concept paper for the gene editing. Okay. But still, this information is only available in the Japanese version, not English version, sorry. Okay, this is, I think, the last slide. So next year, we have an Olympic game in the Tokyo, very exciting. And then next year, actually, this regulatory science strategic consultation is started 7 years ago. The most of the product is already started the clinical trial, and then next year, many product is finalized this clinical trial. So I hope many products to come to the PMDA to conduct the marketing authorization. So, and then, okay. So if you have a question, I'm around here for today or tomorrow early morning. So please catch me, and then if you have a question, please ask the question. Okay, thank you very much.
Thank you. So time is over. So next speaker is Dr. A. Yamada from Angus. He was talking about Japan's first gene therapeutic drugs. Please.
Thank you very much for taking a time and a great arrangement of today's conference. And so I'm very excited to be here and honored to be here to have a little bit presentation of our company and mainly our products, HEF gene therapy product and so on. Excuse me. Okay, today's, excuse me. Okay, this is. Okay. Today's title is Japan's first gene therapy drug, and we are, as you know, the venture company, so we are sticking to the innovation, the globalization, and the today's I will little bit explain our situation and product. So it's a corporate overview. Angus is a clinical stage biopharmaceutical company focused on the development and a little bit commercialization of the gene-based medicine for disease with high unmet medical needs. So skip the other. The pipeline is about fourth pipeline, and the first is HF gene therapy for the indication of clinical ischemia, and today I will explain later a little bit details. Secondly, the N-kappa decoy oligonucleotide for indication of disc degeneration. Phase 1B clinical study is going in USA, University California, San Diego School of Medicine. And the third is DNA therapeutic vaccine for the indication of hypertension. It's a phase 1-2 clinical study going on in the Australia area. And the fourth is naglazyme for the indication of mucopolysaccharidosis and type 6. However, this year we transferred this product to BioMarin in Japan, and already finished the marketing of the product. Again, so this is a pipeline including early stage, for example, decoy, oligonucleotides, and the DNA vaccine, and furthermore the type 2 agonist, and in the case of this, so we have a collaboration with Toronto University spin-out company. Our mission and the vision of the company is to be skipped at this time, so for the time limited. It's a corporate history of a company, and as you know, the spin-out 1999 from Osaka University School of Medicine. And after that, for a long time, so fortunately last year we submitted NDA, and this year Dr. Maruyama explained a little bit of our product today, and thank you very much. And this year approval, and last September we started the marketing together with the Mitsubishi Tanabe Pharmaceutical Company. Basically, strategy is very clear, focus on the novel gene-based medicine, including gene therapy and oligonucleotides and DNA vaccine and so on. As you know, we are a very small company, so not enough commercialization force. So we have, in a sense, we need a partnership with the sales force company for the case of the HF gene therapy, Mitsubishi Tanabe Pharmaceutical, and the other Shonin Pharmaceutical. And we looking for the new seeds for the future. So type 2 receptor agonist and Bioimmunes, Toronto University, and microbiome areas, biotics. It's Israel-based Weizmann Institute spin-out company. And new generation of the genome editing companies. So this company, Emendo, is located in New York area to have, in a sense, a very surprised idea to the genome editing. And the further cancer therapeutic area, so barcode diagnostics, so it means, in a sense, discrimination of the some kind of cancer drug uses. And it's a list of the gene therapy approved in advanced country from the 2012. As you know, the Gribera, the unicorn, and from that product, so at this time, so we could have, in a sense, approval from MHW PMDA this year. And its characteristics of our products, HF gene therapy, the first is, as you know, the Japan's first gene therapy product. And second is a world first plasmid product. Plasmid product is, in a sense, a very classical gene therapy product. However, not yet successful, so at this time, so fortunately, we get it. And thirdly, the world first practical HGF. HGF is, in a sense, a very famous product, and so, however, in this case, first case, and fourth is therapeutic products for peripheral angiogenesis. Many physicians desire the, in a sense, angiogenesis area, so it must be a new market of that disease. Why we select the plasmid? So when we studied the HF gene therapy products, so safety concern for virus vector was raised. So we select the safety way. And secondly, in the case of the HF protein, is administration after that half-life is very short. So as a result, so we select the plasmid vector. Fortunately, so in this case, local and the continuous expression are, in a sense, observed. It's, in a sense, a reasonable case of the application of the HF gene therapy. It's an overview of HF gene therapy for CLI. Substance is plasmid DNA, which encode the human HGF, hepatocyte growth factor gene. Indication of, as you know, the critical lymphischemia, the severest form of the peripheral arterial disease, PAD. Mode of action is the therapeutic angiogenesis. Collagen injected into the muscle in the lower limb. HGF protein is expressed up to the 2 weeks, and after 4 weeks, so plasmid entity is to be disappeared. It's very reasonable to think about the safety. And the transected cell and the circulated entering the activity of the angiogenesis. And again, non-viral vector because of the low safety risk. This figure, upper figure is the pre-treatment. Down figure is after treatment. So as you know, the this vaccination was observed. This is the mechanism of the angiogenesis by HF gene therapy. HF plasmid is administered to the mast cell, and the HF protein was expressed, and the HF protein is to be moved to the old vascular. And the at that time, so and the cellular cell was activated, and the basically as a result, angiogenesis was observed. Its definition of the peripheral arterial disease, PAD, one subset of the systemic SO and peripheral circulatory disorder caused by obstruction of peripheral arteries. And most commonly men aged more than 60 years and senior people. Risk factor is smoking, diabetes, hypertension, and hyperlipidemia. Less pain, ulcer, and amputation cause severe deterioration in QOL. So we focus on the Rutherford classification, focus on the 5 type, and it's, in a sense, ulcer improvement mainly. CLI is, as you know, the very significant and urgent medical needs, especially United States, very large numbers of patients, and more than 100,000 patients per year. And the 1 year outcomes of CLI patients after primary treatment, deaths 25%, continuing CLI 20%. And 2 years after the below knee amputation, deaths 30%, contralateral amputation 50%, and above knee amputation 50%, so very serious disease. It's, in a sense, clear-cut ulcer size reduced significantly. Percent change of the ulcer size versus time course. As you know, the orange line is placebo, and sky blue line is collagen administered, so clear-cut difference, and good results was available. Basically, Japanese development, so fortunately, last year submission, and this year conditional time limited approval, and last month market launch was available. It's, in a sense, as far as we estimate the current situation and conditions, competitive advantage of collagen. So a potential competitor is autologous stem cell therapy. So in this case, requires special facility, and the collection of bone marrow is necessary. And the second is allogeneic cell therapy, so a safety concern a little bit to be considered at these times. Anyway, so as a result of competitive advantage of HF gene therapy, so most advanced product having consistently proven efficacy in multiple trials. And the first in class creating a new products market for CLI, and again, as you know, the low safety risk. Hopefully, HF gene therapy has the potential to dominate the CLI market in the future. It's a last slide of summary. Critical limb ischemia, again, CLI is a serious condition with significant unmet medical needs. No effective therapy for patients without option for revascularization by endovascular intervention or bypass surgery. More than 100,000 cases of amputation every year in the United States only. And the Japanese conditional and time-limited approval and market launching this year, 2019. Next step is we are planning the U.S. clinical study, because as you know, the U.S. market has, in a sense, a large potential. Further, we are looking for the development marketing partner for the territories outside of the U.S. and Japan, so European area or Asia area, so on. Anyway, so we fortunately this year received the approval, conditional time limited approval, and to make a next step for the final approval within 5 years about the PMS. Patient number is 120 plus natural disease course and 80 patient. So anyway, so we are going to, in a sense, do our best as soon as possible. Thank you very much.
Okay, thank you for Yamada-san. Okay, last speaker is Ms. Yoshitsugu from Novartis, talking about the CAR-T therapy development in Japan.
Thank you for your introduction. I'm Yoshitsugu, supply chain head of CAR-T in Japan and Novartis. Before I'm coming to this position last year, I'm leading the CAR-T development in Japan for 6 years. Then today, I'm very happy to share our experience. Before I'm moving to Japan development, I'm sharing Novartis CAR-T therapy, manufacturing, and supply chain. First of all, let me introduce our CAR-T experienced patient. He is the first DLBCL patient treated in 2015, and she is the first pediatric AL patient treated in 2012, and she is very famous, and she is coming to this symposium on Friday. I'm very happy to meet her. And there, our hope, and Japan team encouraged there, with one infusion, their clinical benefit is quite long for several years. Then to bring this CAR-T into the Japan market, Japan team had worked a lot. This slide shows the Novartis journey of bringing CAR-T globally. Blue parts are related to the Japan activities. Before entering the collaboration with the University of Pennsylvania in 2012, Japan team initiated the development. And 2015, we submitted IND to 2 indications. And 2016, we obtained orphan designation. Then after the first approval of CAR-T in US for pediatric AL, we filed an NDA in April 2018. After the many countries' approval, EU, Canada, Swiss, and Australia, we finally obtained approval in 2 indications in March this year. In 2 months later, Japan Health Authority decided the reimbursement for our CAR-T. Our CAR-T is autologous, and after the blood collection from the patient, the T cells are genetically encoded by using a lentiviral vector. And this CAR-T cells works in the body to recognize and fight cancer. And also, the CAR-T cells can expand in the body. That's why the CAR-T is called as a living drug. With this, only one infusion can bring the long-term benefit. Our CAR-T is utilizing cryopreservation in manufacturing. The benefits are apheresis scheduling flexibility, and we can avoid the risk of transportation delay, and we can preserve cell quality. And our CAR-T is using 4-1BB as a co-stimulating domain, as well as antibinding domain, anti-CD19 domain, CD3 zeta signaling domain, and also CD8 alpha hinge and transmembrane. 4-1BB could enhance cellular expansion and persistence. And our CAR-T was studied in global clinical trials in 2 indications. One is pediatric and young adult patients, and another is DLBCL, diffuse large B-cell lymphoma. And these studies are conducted in 10 and 11 countries, including Japan. I want to briefly mention about the clinical result from pediatric ALL, Iliana. In 24 months follow-up, the complete remission and complete remission without the blood recovery was observed in 82%. And at that time, duration of remission and overall survival were not reached. This means that CAR-T is quite durable and sustained, and CAR-T could be a definitive therapy for many patients in this population. And this slide shows the Juliet study's result for DLBCL. In 24 months follow-up, overall response rate was 54%, including 40% CR. And if patient achieved CR, and response was quite long, and 70% of the patients were not progressed at this time. We could see the toxicity, including cytokine release syndrome and neurological events like this. Now I'm going to CAR-T manufacturing and supply chain. Kymriah has a sophisticated manufacturing process. First one is, leukapheresis. To collect the blood from the patient, we are doing leukapheresis, and these T cells are preserved at the site. To secure the quality, we are doing the audit and certify the site, like other raw material vendors under the QAA, Quality Assurance Agreement. Then this qualified T cells are moving to the manufacturing facilities, and T cells are enriched, activated, and encoded genetically via using lentiviral vector. Then CAR-Ts are expanded. Before the release, strict quality testing is done to secure the high quality. Before the cell infusion, lymphodepleting chemotherapy is given to the patient to reduce the white blood cells in the body, which helps the CAR-T works in the body. And CAR-T needs multiple treatment center stakeholders' involvement like this. To give the CAR-T successfully, many stakeholders' involvement are needed in the hospital. So this slide shows the supply chain for CAR-T. After the patient is identified, we will receive orders, and all scheduling is done via electronic system. With the hospitals, we are scheduling apheresis, presentation, pickup time, and the delivery time. And with the transportation vendors, we are scheduling the pickup time, export, and import, and deliver to the site. At the transportation, we are using dry shipper under the liquid nitrogen dry paper phase condition. Of course, we need a communication with the manufacturing site in US, and also we have to communicate with local release testing site. Since Japanese health authority requires us to do local release testing in Japan for commercial products, then we have to communicate with local release testing site. After that, finally, this product is shipped back to the hospitals. The dedicated team is managing this whole system in each patient, and we are always closely monitoring this part. So this is the last slide to explain this flow. This slide shows the importance of the chain of identity. Since our product is autologous, to maintain the chain of identity is quite important. To do this, we are using these information: patient name, date of birth, batch ID, and apheresis ID. Since Japanese Health Authority requires us to use the Japanese patient name in the final product, we have decided to use a combination name, Japanese and English, as a chain of entity information, because this is the Japanese regulatory requirement. Also, we are using US facility, and US facility needs to confirm the name in English. Then in each step, these COI information has to be confirmed. Self-correction, characterization, and shipping, manufacturing, and the delivery at the site. Now I'm moving to the Japan development. This slide shows the pathway to the IND. And the PMDA has set up several consultation schemes toward initiation of clinical trial. But these consultations are not mandatory, but some of them are strongly recommended before the IND. We did several preliminary meetings with PMDA before the IND, and we did 2 formal consultation meetings. One is regulatory science strategy consultation meeting, and other is consultation for clinical meeting. At the RS strategy consultation meeting, we discussed preclinical safety data and CMC quality, including specification of test method of clinical product, compliance with Japanese standard for biological ingredients, and applicability of Cartagena law, such as Maruyama-san mentioned. Then in our case, the Cartagena is not applied because we could show that no viral exists in our CAR-T. And no regulation for import, export to raw materials and final products because we are using US facility. At the consultation meeting for clinical, we discussed endpoint, target population, Japanese patient number, dose and schedule. During the clinical trials, we discussed many topics at the preliminary meeting, including definition of starting materials, manufacturing license certification, and necessity of GCT inspection in each facility, and product composition, which is necessity of kit for local apheresis. At the PMDA consultation, we discussed adequacy of data package and Japanese patient data. Although the PMDA recommended us to do quality consultation before the NDA, we did not do from the timing perspectives. After the NDA, we have a lot of communication, inquiries, and answers with PMDA, such as in the CMC, master file registration for raw materials, verification and local release testing, and also clinical efficacy and safety. After the robust discussion, we finally obtained approval for 2 indications in this March. In addition to actual CAR-T preparation, we have to consider the necessary materials and drugs for related to CAR-T, like this. At the local apheresis, we need local apheresis kit and ICDA. At cryopreservation, cryovac and cryomedium are needed. Before the infusion, we are doing lymphodepleting chemotherapy, which includes fludarabine, cyclophosphamide, and other drugs. And at the CAR-T treatment, infusion set is needed. Then after the CAR-T infusion, in our case, adverse event is observed. Main adverse event is CRS, cytokine release syndrome. To manage CRS, we need anti-IL-6 agonist, tocilizumab. So Japanese Health Authority requires SNDA for LD chemo and tocilizumab. And after many discussions with other companies and health authorities, SNDAs were submitted, and these drugs are approved as an indication for preconditioning therapy of CAR-T and also CRS treatment by using our CAR-T clinical trial data. In addition to that, we discussed about a kit to collect raw materials. During the clinical trials, we provided cryovac and cryomedium because at that time they are not available in the market. But after that, we decided to use available kit, available vac and medium in the market. But PMDA said to secure the raw materials quality, we might need to provide these kits. But we decided not to provide these kits. On the other hand, we decided to contract with the site under the QA, quality assurance agreement. To do that, we are securing the quality of raw materials. Now I'm sharing the current discussion about OOS, out of specification product. This is not only related to development, but in the commercial setting, we have to discuss. We are doing release testing, but sometimes the specifications are not met. In this case, this product is regarded as an OOS, out of specification. And we can't provide OOS product as a commercial product. However, the treatment options of CAR-T are very limited and urgency of treatment is high. We have to consider to provide OS product if this is ethically desired. Then Novartis is globally considering to provide the OS products under the scheme of each country. In Japan, OS product is regarded as unapproved, and we can only provide the OS products under the scheme of industry-sponsored clinical trial. But I think this needs to be more discussed with health authorities. Lastly, I want to share the scheme to collect real-world data. Post-marketing surveillance is required in Japan, and in our case, we have decided to use the database at academia, Japan Data Center for Hematopoietic Cell Transplantation. The database survey is conducted as PMS. This is a flow to collect data at database survey. The hospital input the data into the database, and this data is providing us under the scheme of agreement. Also, we are making an agreement with the hospital to collect the data. And we believe this scheme could be used for future CAR-T therapies in Japan. So, although we are in the stage of commercialization, there are many challenges here, such as understanding to regenerative medicine, expansion of manufacturing capacity, how to treat OS products, patient selection, and promotion of appropriate use, and collaboration with academia, cost with long-term data, and international harmonization of regulations. We want to overcome these challenges by the collaboration with many stakeholders for future CAR-T success. So finally, I want to thank everybody who are involved in the CAR-T development in Japan. So thank you for your attention.
Thank you. This is on time. So session is closing. So we hope this session's presentation would be helpful for you, your industrialization, and our near future collaboration. Thank you.